Hello everyone, my name is Simona Balestrini. I'm a child and adult neurologist. I work between Italy and the UK.
My name's Rhys. I'm an adult neurologist. I work in Newcastle in the north of England, and I particularly look after people with intellectual disability and epilepsy.
My name is Moore-Ramdin. I'm the Global Medical Director at Jazz Pharmaceuticals. I'm a pediatrician with the neurology interest.
Hi everyone, I'm Yvonne Goodson. I live in Oxfordshire with my husband Mark. I have two daughters, Verity, who's not here, and Lydia, who you've probably met.
So perhaps I can start. First of all, thank you everyone. This is a really great meeting and very inspirational also for us as clinicians, I think I can say. We learn so much every time. And so the focus of this panel is treatment. And we also look at some of the questions that came from some of you. So the idea is to try to address some of the questions. And I will start with addressing gene therapy, which is one of the novel treatments emerging for some epilepsies. But perhaps before talking about gene therapy, I think we should go back to the concept of precision, which we heard a lot throughout the day today, because what we are learning more and more really in the epilepsy field, I'm also focused, at least my research and clinical work is focused on genetic epilepsies, is this kind of new paradigm where we try to understand exactly what is causing the epilepsy, but not just the epilepsy, all the other aspects, because we are really trying to now go beyond the epilepsy. And once we understand the cause, then we can try to be as precise as possible to address the cause. So that's the kind of concept of precision. And so in Ring 20, as David explained greatly earlier, is more difficult because we don't always understand exactly what's happening. So we don't know what are, for example, precisely the genes involved. We have some potential hints, like those two genes that we heard earlier about. But then we don't know exactly how those genes are involved. We don't know if they are involved in the same way in all people with Ring 20, for example. And so that's how it becomes more difficult to be precise. So in general, the use of precision approaches in epilepsy is still quite limited, because even when we know exactly what the cause is, sometimes we don't have some treatment to address that cause. Or sometimes we do have that treatment, but that's not easy to access. Or sometimes we can access it, we use it, and it doesn't work. So we have all kinds of possibilities. So what we do still in our clinical practice is, first of all, to really try to be precise, at least at the individual level, because every person with epilepsy is different. So we really try to understand the person holistically. So we need to look at all the problem. And I think that quality of life should be always the first hint, because even when epilepsy is very severe, as Lydia very well said earlier, we can't just focus on giving lots of anti-epileptic drugs to control the seizure, but then, for example, the person is sleepy or cannot really work in the daily activities. So we need to find a balance for each single person. So that's what is about precision. Regarding gene therapy, what is being developed, although it's still very much at the kind of experimental level, so there are lots of animal models, for example, is try to repair somehow the gene defect. But that implies that we know exactly what the gene defect is. And for example, there are some bits of DNA that can be used to change the gene activity. Because, for example, in some cases, we know that that the gene contains all the information to build a protein, and that the protein is that molecule that kind of is responsible for the function of each single cell. And so if there is a change in that gene, that the protein won't work. And so this new kind of gene intervention can try to somehow modify the gene expression. So if the gene is overworking, that try to kind of reduce the gene activity, or vice versa, if we know that the gene is not working properly or there is too little of that gene, they kind of increase the activity. So this is a kind of approach. But there are a lot of but there, because first of all, we need to be able to safely deliver a vector, for example, to put the new DNA into a body. And that's not always easy. Viruses are used, but of course to bring a virus into a brain is not the easiest way. Plus, we also need to know exactly what is the defect, because we don't always know. Even if we find a gene change, then sometimes we can not link that gene change with the abnormal activity. We don't know if, for example, the problem is that the gene works too much or works too little, for example, and we also don't know the interaction with the other genes. That's why I also think that what David told us earlier is very important, because really we shouldn't stop at ring 20. We should look at the whole picture, the whole background, because even if there is this abnormal conformation of one chromosome, we need to know what happened to all the other genes, because all the genes really work together. I mean, our body is so complex, our brain even more complex, so we need to know the interaction. And for example, we are doing this on a research basis for another genetic epilepsy, which you heard also earlier about, which is Dravet syndrome. There we know probably much more compared with ring 20, because it's less rare and because it's been more studied. But even there, we see lots of variability. And even there, even if we have a bit more precise treatment, most of the time they do not work. And so there we are now looking at the genome. Even if we know exactly that what is the gene involved, we know that the gene in most cases lose the function, but still we are looking at the genome and we try to understand how the other gene influence that specific gene change that we already know about. And I think probably for ring 20 as well, that should be the direction, because if we are able to characterize exactly the genome of each individual, then we know much more. And so, I don't know, at least my vision is that probably in the near future, a genome analysis should be a bit like an MRI that pretty much every person with epilepsy has, because it really helps us to get the full picture at the individual level, and that should help us then to tailor each specific treatment and going beyond seizure treatment, but also try to address the risk of cognitive decline, behavioral problems, and sometimes also other organ involvement, depend of course on what's the kind of genomic background. So that's about gene therapy. Now, I don't know who like to go next. I think probably from what Alison suggested, we could have Yvonne and Cindy thoughts about treatment.
School, I'm afraid, guys. Paper. Alison has asked me to tell Lydia's story. On the slide is part of this story. It doesn't include the many hospital admissions and a couple of ambulance trips. Neither does it include the dealings with the education authorities, social services, or the Department of Works and Pensions. Everything feels like a battle. We are incredibly lucky here in the UK. We have the National Health System Service. And as you can see, we have been to many hospitals and seen many people. With the odd exception, everyone has been kind and as helpful as they can be. When Lydia was 4, she started having what turned out to be seizures. She called it the plinks. It took 6 months to get diagnosed. Yes, you did. I can remember. 2 years off meds and she appeared to be fine. She was happy-go-lucky, popular child. During this time, I had her hearing tested twice. Looking back, I guess she was having brief, non-convulsive seizures and wasn't deaf, as I thought. When Lydia was 11, the seizures returned. It didn't take long to get the ring 20 diagnosis, unless you count from when she was 4. And we started working our way through all the meds and the side effects. When we started on the meds, the seizures skyrocketed from a couple of weeks to one every 40 minutes at the worst time. Also, there are the side effects to deal with. On gabapentin, she never stopped talking. Not talking to anyone, just talking. We called it gabapentin. Leviracetam brought on Keprorage, which is horrible to deal with and I'm sure to experience. On clobazam, Lydia was practically comatose, gained half her body weight again and had compensated hypothyroidism. She was difficult to wake in the mornings. I used to sit her up in bed, wrap her in a dressing gown and use that to drag and bounce her down the stairs. At age 15, Lydia slipped into non-convulsive status. She was half in and half out for several months. She was admitted to the high dependency ward in the John Radcliffe Hospital, pretty much at the insistence of one of the A&E nurses. Lydia was in and out of the JR several times over the next few months, during which time she had a gastrostomy button fitted and was pump fed. This would have been summertime. In the October, she was having a pump feed. She looked across at the carbonara I was making, homemade, not from a jar, and I asked her if she would like some. She nodded. I gave her a serving, and after she started speaking. This was the first time she had spoken for 4 months. Since that time, things have been fairly stable. However, 18 months ago, Lydia had another hospital admission due to non-convulsive status. There followed a series of mix-ups and wrong blood tests taken. Her meds were adjusted, and as a result, she went into toxicity, which wasn't picked up quickly. This resulted in a deterioration in her walking. To be honest, I can't remember half the stuff we put Lydia through. She's a tough, brave young woman. Parts of her story will be familiar to most of us here, so I won't go on much longer. Our neurologist, Dr. Pike, asked me after one of the med changes if things had improved. I said no, just another load of not good enough. Thank you for listening.
So back to the doctor perspective with some more hope on alternative treatment maybe.
Yeah, thank you very much. First of all, it's really humbling to be with you today. Really humbling to hear the stories, and thank you very much indeed. And a lot of what I'm going to tell you, Lydia's already said. You did an amazing job. You did an amazing job. So doctors probably don't know much about Ring 20. That's the first thing. And if they know a little bit, they may not have seen a case before. What they've picked up is from a book or an online journal. And what they will have read is that it's hard to treat. And sometimes that makes people think, well, nothing works. And therefore people might expect some side effects for patients with Ring 20 they wouldn't expect with other things. They might push the medicines too hard, too fast, too many changes. You change the medicines, you end up with side effects. It's a really stormy course. It's a really stormy course. But just because it's hard doesn't mean you've got to accept things to be second best. And there's a lot that you can do to get the most out of your neurologist. And the first one is find somebody who will listen to you. They don't have to be an expert. Experts are great, they probably get there because they do listen, but somebody who listens to you, or somebody who's got a great specialist nurse or a great team, somebody who will call you back if you pick up the phone. Prepare for the consultations. We love video. We love video, particularly for Ring 20, because we miss so much. Little seizures from sleep. Are they seizures? Are they not? They're probably seizures, but and the non-convulsive status. We miss that so much. And so video is helpful if we can't get a hold of EEG. The previous medicines are important to keep on top of, otherwise we'll say try something again, try that Keppra again, what was the problem again? You go, never again, no, give it another go, so never again. And you're right, lots of patients come to our clinics and say we've tried everything, and they, oh, I bet you haven't. But for people like those with Ring 20, they probably have been people who've always been trying, and yet not always first in the queue for medical trials either. They seem to sort of miss out on some of the medical opportunity that comes with studies as well. So what do we currently have? We currently have all of the tools, medicines and non-medicines that Lydia mentioned, that we've borrowed from the other epilepsies. So we've got probably 30 anti-seizure medicines in the UK that we could use, of which I'd probably ignore the first 5 or 10 because they're so old-fashioned and so dirty you'd never use them again. And then you've got a combination of new medicines that are a bit stronger and some medicines that are a bit milder that you're trying to get lucky with, because there's nothing else that we can tell you in terms of where you're going to be. And that's probably why we've stumbled across medicines like valproate and lamotrigine on that list in terms of trying to get that balance. Almost all the medicines, if we use too much of them, are going to make you drowsy, and drowsiness and seizures can make behaviour worse, and so behaviour is that third element with the seizure control. So we look for things that don't necessarily make you drowsy. The ketogenic diet's one of those. Vagus nerve stimulation is one of those. Surgery for some people is one of those. But if you came to my clinic, even though I'm an enthusiast and I'm in a well-funded centre, as an adult I can't start the ketogenic diet in England. You know, there are only a couple of centres that can, so I can't do that. I can put a vagus nerve simulator in, that's easy, but there are other centres elsewhere, maybe not in the UK, that can't get access for that. Reimbursement for VNS in the States is hard. And so it, again, is another thing that's not fair. One of the things that can help is helping to frame the epilepsy differently for your neurologist. So people say it's ring 20 and therefore we can't do that. It's ring 20 so we can't give you cannabidiol, can't give you Epidiolex. Well, actually, if you deconstruct the elements of it, so in the United Kingdom, the licensing, so the rules I have to follow for the cannabis medicine is seizures associated with a certain type of epilepsy that we call Lennox-Gastaut. Doesn't mean it has to be Lennox-Gastaut, it has to be seizures associated with. And so that's how we prescribe cannabis medicines for people with ring 20 and other conditions like it. Because my role isn't necessarily to be a gatekeeper for medicines, my role is trying to get the right medicines to the right people. I'm going to stop there because what I'm going to say isn't more important than Yvonne and Cindy, and the questions come later.
Yes, oh God, I hate looking at my picture. Anyway, many asked when I left pediatrics, why would you move to the dark side? What's wrong with you? And many reasons. I think, you know, as clinicians, in medical school, you're told you have to be a clinician, you have to deal with patients directly. There's so much that we can do outside of that to really help people without necessarily seeing them face to face in a clinic. And I've been so impressed by the people I've worked with cross-functionally, whether they're in marketing, in research, in commercial development, that really care about what happens in this sort of arena. And that's why I'm still in the job after 15 years. And I think what is happening now is a real shift in pharma to start listening to what the medics within the company have to say. And it's meetings like this that help me bring that message to the CEO and others who are looking for the return on investment. Let's remember they're not a charity, all right? They have invested almost $1 billion or more to bring one molecule from concept to, you know, to Rhys and, you know, to the patient. And, you know, you have to find ways of recouping that. And one way of recouping that is finding new diagnoses or new challenges that patients are experiencing for the company to actually say, okay, can we repurpose a drug that is already licensed for something else, or do we develop something new? And it's the science that will drive that. So it always comes back to the science and listening to patients and what is their most challenging issue. One paradigm shift that's happening in the work that I'm doing right now is looking at non-seizure outcomes. Because that's a key element that I think the regulators are struggling with at present. They insist that we have to do research looking at the seizures and showing efficacy in seizures. But it's a lot more than that. And I've worked enough in pediatrics to understand that it's the behavioral issues and the cognition problems and not being able to go to school and the aggressive behavior and anxiety and all those elements that really make your life as a parent, as a family member, really challenging. And so I'm really hopeful that even though we may not have the anti-seizure medicines that stop the seizures completely, that there's hope for showing the regulators that there are other things that these medicines can do, and some of these medicines may not have the side effects that the old preparations may have. So we're doing a lot of work with the NHS and also with the research foundations and also with the FDA and EMA right now to understand, okay, what studies, what research do we have to do to prove to them that these drugs that are already present or that we can develop can be approved for, you know, the symptoms you see in ring 20 and in all other rare epilepsies. And again, I echo what Rhys says, it's sometimes it's not just about the syndrome itself, but what are the types of seizures that we could, you know, justify using the medication for, because not all patients are the same. Yeah? So lots of work to do.
Thank you. So I think we could probably have now some questions from you directly here, and then we can probably try to address some more that came before. Alison.
Can I ask two very quick questions, one for Dr. Rhys and one for Dr. Lisa, please? So my first question to you, Rhys, is when you spoke about LGS like epilepsy, and one of the challenges that we see amongst the community is the ability for their doctors to prescribe them drugs that are licensed for LGS-like symptoms. Some in our community have prescriptions for those to try those medications, whilst others, their doctors absolutely refuse, and yet they all have a Ring 20 diagnosis. So there seems some inequity in prescribing there, and it's subjective on behalf of the doctor who is treating you, which seems really, really unfair, because doctors are quite happy to treat our children on a trial and error basis with other licensed anti-seizure medications. 3, 4, we saw anti-seizure medications together at the same time, yet who has done a clinical trial on those 3, 4 medications at the same time? So that to me seems unethical, but that's a challenge. So that's my first question. And before I forget, my second question that will go to Lisa is, in terms of clinical trials, what do we need to do as Ring 20 and the other epilepsies outside of the bigger, more common rare epilepsies such as Dravet syndrome we've heard about today and LGS, where all of the new and emerging new anti-epileptic medications seem to be trialled in? It's those patients that get access to those therapies first. What can we do to encourage pharma to widen the opportunity to a wider group of epilepsies, including Ring 20?
Shall I go first? It's not fair, is it? It's really not fair, and particularly when you sit as you do and you have these stories coming in from different families who ostensibly have got children with the same epilepsy and they're getting different treatments. It's not fair. And the second thing you said about it not being fair is people who experiment with different drugs, different indications and different doses. And I guess we're really talking about cannabidiol, we're talking about Epidiolex mostly here as one of the inequality. So within the UK at the moment, when I prescribe Epidiolex, the cost is taken up by NHS England. So it doesn't come out of my budget, doesn't come out of my hospital budget, which is good. The bad side of that is I'm prescribing within a framework. It initially felt like a straitjacket, and there's almost nothing else I prescribe that has that same straitjacket around it. And I've got a ticker box saying, this is Lennox-Gastaut syndrome. And I think that feels so strange and so odd for UK doctors that they've become very restricted in their practice. And that's if you ignore the fact there's more paperwork for them, so maybe they haven't even gone down that route in the first place. Nowhere on that form have they defined what this Lennox-Gastaut is. Only this year have the powers that be given us a Lennox-Gastaut framework to work within, and that framework does not work for adults. It works for kids, but doesn't work for adults. Works going forwards, but if you try and go back, it falls to pieces. So I would still say that falls down to the individual doctors who should be making a best interest decision for the person in front of them, and that's where I prescribe for Lennox-Gastaut-ish, and I think that's fair enough. And in doing so, I've never caused, I think, anybody any harm. The parents understand what's happening. It's an easy conversation, and they'll say yes, no, or maybe, or not now. But I think you can prescribe in that way. I think it's defensible, because this isn't a clinical study where I'm going to ruin Lisa's results because I've given somebody with proper Lennox-Gastaut medicine and somebody with Lennox-Gastaut-ish medicine who will never know what works. This is about, is somebody going to be better by Halloween or Christmas, or keep them out of intensive care? And there's an education gap, particularly with adult neurologists there, that we need to fill.
As to your question, I think there's a two-pronged approach. I think pharma realizes that we can't do anything positive without your support. And I think it works both ways. It's definitely a symbiosis, right? So within Jazz and within many companies now, we have patient advocacy leads who interact with the various groups and bring that information in, like what I'm doing now. But the other thing is that I found really helpful in past experience, trying to get approval for a drug for a certain syndrome, for example, is the patient groups actually lobbying directly to the regulators. That's such a powerful thing. It's much more than I could ever bring to the table, because they're listening to real cases, And they have to get over this numbers game that we're always playing, how many patients we have to treat to get this effect. And I don't care about the numbers. All I know is that my cousin has gotten loads better because of this drug. I want it for him. So what we're doing now is working a lot closer with the MHRA, for example, in the UK, to try and understand, okay, what mechanism is there for us to do a small study, almost like an N of 1 project, where you have 1 or 2 patients with a very rare syndrome, you try the drug on them, measure them, make sure you understand what's happening over several months, and present that data to the regulators and say, okay, would you pay for a small cohort of patients, almost as a trial run, and see how that goes over the next couple of years. And that in itself will be proof of concept to think about approving the drug for a much wider population. So we have to sort of take it in stages, but I think your voices is probably what's going to move the needle more than us, because we think we know, but you know better than us, really. And I have to depend on people like Rhys and others to fight their corner within their clinics, because if you have 1 or 2 patients who are responding, then we could use that data within pharma, within our submissions, to get the regulators to change their thinking.
For Dr. Balestrini, have you found when you're treating kids with Ring 20, we happen to notice that when people submitted their meds, that were a fair number of kids on either an ADHD medication or an antidepressant. Have you found those to be useful in this population? I know it's a small number, but I thought I would ask.
Well, I think, again, it really depends on the individual presentation. I think when a child has a prominent behavioral disturbance, difficulties with the attention, with the behavior, to a level where the functioning at school, for example, is compromised, then definitely they can benefit from specific ADHD treatment. And actually that in turn also improves the epilepsy control. So definitely I think, again, it really needs to be judged from the single person, for what are the prominent symptoms. And that's when, again, it's really fundamental, the discussion with the family, because we really need, as Rhys was saying, it's fundamental to listen to all the single concern and try to kind of decide together what's the best scheme of treatment. And it will always be somehow a try and error process, because we may prioritize attention difficulties, but then kind of seizure may become prominent at the second stage. But yes, the answer is yes, but it needs to be judged at the individual level.
Question, I was very much, I'm every time very much surprised when talking to other parents, but also seeing this slide that was presented with 15 or 18 different drugs. And then I wonder, does that come from purely everybody is different, or does that come from starting with something, adding another drug, then taking one off, adding another one? That doesn't work, but it might not work, that drug, because of something else, or what you tried before, or you build up, wean off too quickly, and I think that's almost driving everybody crazy.
Yeah.
And it's not working.
Yeah.
It's only 2 bases that are wrong.
Could you imagine something different? So imagine everybody with a complex rare epilepsy is put into a registry, international registry, and therefore every trial and error that we've used is tallied up against quality of life, counting seizures and other things that are important. And then if there is a signal of these medicines work well in combination or don't work well in combination, we would find out at the earliest possible moment, rather than stumbling blindly, potentially for decades, and causing harm, because we will cause harm. With medicines, there'll be times where the medicines are misprescribed, my letter's wrong, people take it incorrectly, the hospital get it wrong, so they're looking at old letters. Yeah. Data, connectivity, families, bringing things together.
I absolutely agree with that. But the challenge for us as a really rare patient community is we've seen today that we've come together, it's taken us 5 years to fund a 1-year natural history study and start a patient registry.
Yep.
But we can't sustain funding with 120 families across the world for the millions of pounds worth of studies, for the sort of data collection that you're talking about. We need the support of pharma, of other industry, biotech, to come on the journey with us, because we can't fund the tens, the hundreds of thousands of pounds, dollars, euros that requires to give the basic information, to get the scientists interested, to get pharma listening and looking at us, to get the clinicians, to get them the information that they need to treat our children. We can't do this alone. So my call out to all of you is, please help us, because we can't do this on our own. But we're willing to do whatever we can for our children.
Thank you.
I have I'm not sure whether you want.
Yeah, no, please go.
Two other questions. So first, Catherine, she said, not sure if this can be answered or not. Can Ring20 cause physical manifestations or mimicking other conditions such as stroke episodes, random paralysis, etc.? When things, I know my notes went away.
Sabotage.
But the chat is gone. Who killed the chat? Oh my God. Yeah,
So differential diagnosis, well, probably Rhys will have something to add, but I think it's not always easy to arrive to the diagnosis. As you know, there is always some delay between the clinical onset and the diagnosis for probably a number of reasons. The presentation can be variable, and especially non-convulsive status is not always a classical seizure presentation. So neurologists need to know it. I mean, as Rhys said, some neurologists probably don't even know about the syndrome. And it would be really important to have an EEG, because some symptoms may be very non-specific, may not remind a clinician of kind of epilepsy activity, in which case, yeah, it could be, if it's a kind of very acute onset, it could be kind of stroke-like, or people may think about other things. And the second aspect is probably the difficulties with the genetic diagnosis that we had earlier. So people may have the full exome or genome sequence and still not found the ring conformation. So I think that adds another layer of complexity to achieve the diagnosis.
yes, and then to add back to the chat, to add to that, yeah, definitely if these things don't happen at hospital, knowing that we are 99.9% hopefully not in the hospital. And then, yeah, one additional comment was, and then doctors say that it's psychiatric.
Yeah. So there are a couple of things that are, Ring20 is not unusual for giving us this problem. It's just that with Ring20 you've got non-convulsive status, which is really hard to know, are they in or are they out? Behaviour, side effects of medicines, seizures from sleep, and some are frontal lobe seizures, which themselves can be a bit quirky, and it's difficult to know what's going on. Put that as a package, and I'll just say that's tricky. That's really tricky. But what we're looking at in the next couple of years are better monitoring of people outside of hospital. I'll give you an example of two that would be useful for Ring20. One is the use of high-resolution fixed camera video, normally in a bedroom, to count motor seizures. And they use AI to tell you what's seizure, what's movement, what's rolling over. And the company puts that together and sends that then off to the neurologist saying, do you know what, the family report 8 different types of events at night. We've put it together and there's clearly seizures and rolling over. And actually we're missing a lot of seizures. So that's a bit of kit that's being studied in the UK at the moment. And then the second is what we call long-term wearable EEG, or ultra long-term, which is a wearable device. It can stay in for a year, probably 5 years. They just don't have it kite marked for 5 years. And you may not necessarily want something behind your ear all the time, but if you're going through a bad period, you touch the battery, stick it on, and it would tell your neurologist, you know, this is seizure, this is psychiatric, this is sleep. And so for people with autism, intellectual disability, complex seizures, I think that's going to help us unpick a lot of that 99%.
Do you have any experience, positive or negative, on using pharmacogenetic tests we're trying to find the most appropriate molecule for your patients according with the haplotype on cytochrome or other genes involved in metabolism?
That's a good question. So, well, pharmacogenomics is kind of expanding field in epilepsy. I think that we probably should distinguish what we know in terms of the level of evidence. I think for some pharmacogenomics marker, there is robust evidence. Classical example is a HLA marker, which is just a kind of a type of genetic marker that we all have, but some ethnic groups have a more common prevalence of specific type of this marker, and that predispose them, for example, to a very severe allergic reaction to some drugs. So we know in our clinical practice that if we have, for example, a patient that comes from some South Asian region, then we should first do this kind of testing before trying phenytoin or carbamazepine or specific type of drugs. So that as a high level of evidence that is already implemented in our clinical practice, for example. And then there are lots of other studies that unfortunately were not really replicated, so we don't really know how robust they are, and so we can't really use that in clinical practice, but we do know that, again, every person is different, for example, in the way they metabolize the drugs. So for some, for example, benzodiazepine, we know that some people are more kind of slow metabolizers, and so they need lower doses, otherwise the drug is kind of get too high level in their body, and then they can get the toxicity such as too much drowsiness and so on. And then on the other hand, there are people that could be kind of faster metabolizer, and then they need higher dose. So we kind of have some evidence. It's of course all genetically determined, but we don't do pre-genetic testing before deciding how to treat those patients. So that kind of pharmacogenomics that informs about the risk of drug reaction, I think it still has a limited value in our clinical practice, except for those examples that I just said about specific ethnic groups. And then, of course, there is all the other chapter of pharmacogenomics in a way that is knowing the gene that is responsible for the epilepsy, because if we know that there is a single gene problem, that can also inform us, as we discussed earlier on, ideally some precision treatment that go to reverse or correct that defect, but also sometimes can tell us what drug not to use. And again, probably we should take the example of Dravet syndrome, where we know that the problem is with the sodium channel gene. That sodium channel gene doesn't work properly, and we know that if we use a sodium channel blockers, and still most of the drugs we use as a kind of action on blocking the sodium channel, that those people get really worse. I always make this example of this 6-year-old patient that had the diagnosis of Dravet syndrome very late. He could not speak, he had very terrible seizure with convulsive status 2 or 3 times per month, very severe. And he was on lots of sodium channel blockers because he didn't have a genetic diagnosis. When we made the genetic diagnosis of Dravet syndrome and we removed the sodium channel blockers, this patient for the first time started to talk again, which the family thought it was a miracle. And he stopped having convulsive. He still had seizures, and of course his cognitive impairment was still kind of moderate to severe, but there was a dramatic improvement. So, sorry, probably I had a too long answer, but pharmacogenomics can inform us a lot in our kind of clinical practice with different, in different aspects, but still there is not probably a kind of consistent paradigm we can apply to all patients. We still need to really act on an individual basis.
I'm really sorry, but I think I'm going to have to call it a day because it is 5 o'clock. So I want to say thank you ever so much to the panel, and I hope that you might get... I have drawn the short straw because I have to talk about the whole day now. And actually, yeah, please have a seat. I'll just do I had to get my crib sheet out really because it's been such a full day and so many interesting presentations that remembering what we did in the morning and then thinking about everything that happened in between and coming up to now is quite a challenge. We've learnt, I think, well certainly I have learnt a tremendous amount today, and I hope that that's true for a lot of us, about the condition, about the current state of research, about genomics, about treatments, and so on and so forth. And I think it's just been a remarkable day. I hope that those people who've joined us online have gained a lot from it as well. I know it's a lot more difficult if you're watching it on a screen, and it's been really wonderful being in the room, and you know, you get the vibe as much as anything else, and the contact between families, clinicians, and everybody else. So it's been a wonderful day, and I think what we would like to do just briefly, so two things, to say thank you to our speakers. That's the first thing. The second thing is to do a group photograph, which we do want to do. Are we doing it in this room?